Hello, I will be presenting my project work as a part of my course Element of Synthetic Biology. My topic is Solution to Non-Renewable Resources Using Synthetic Biology. My name is NISHANT VAJA and my ID is 2014-01-183. So as my groupmate explained the procedure of converting biomass to biofuel, in that procedure biomass like sugarcane, corn can be used, which are also a food. So there will be competition for such biomasses, whether it should be used as food or biomass. But in this method of converting biofuel from biomass, algae is used, so such problem of feedstock will not be there. Now I will explain how we can get biofuel from algae, and in the end I will explain some changes which can be solved using synthetic biology. So basically, the consumption of non-renewable resources like fossil fuels are higher than production of them. As we can see, it will lead to depletion of non-renewable resources. So we have to get solution of this. So we can take an alternate way of using fossil fuels, or we can either increase production rate or decrease consumption rate in such a way that consumption rate is less than production rate. As far as alternate way of fossil fuel concerns, we can produce biofuels from algae. When people use this fossil fuel in their vehicle, it produces some toxic gases like carbon monoxide, which are not good for the environment and health of people. But when the biofuel, which is derived from the sources like algae, is burnt, it produces carbon dioxide, which can be removed from the environment using photosynthesis. So environmental benefits are also there for using such biofuels, and algae is also biodegradable, so it won't affect the environment. When biofuel is produced from the crops, it requires the land and water and lot of other things to grow the crops like sugarcane. But in the case of algae, it can grow into wastewater. So this is one of the very important characteristics as world is facing problem related to freshwater. Producing fuel from algae costs more than producing fuel from crops. But yield of fuel from algae is 100 times compared to fuel derived from the crops. Idea to derive fuel from algae is basically extract the lipid or oil from algae and then process this part to get the different fuels. These techniques are one of important thing which determine this success of procedure. Synthetic biologists should work here to develop various efficient techniques. Here, if we want to rely on the biofuel procedure by the algae, then production of algae should be at the rate which can overcome the consumption rate of fuels. There are two main ways which are used to grow the algae. One of them is photobioreactors. Photobioreactors are also called PBR. One of the main features of the photobioreactor is one can regulate the different things like temperature, carbon dioxide, etc. So user can decide the optimal quantities of these attributes so that algae can grow very fast. Other main usage of PBR is one can avoid the contamination of algae with other species which resist the process of converting fuels from algae. Since PBR is closed infrastructure, so the evaporation of the water will be less and loss of water and usage of water will be significantly less. In this PBR, because of lesser carbon dioxide, the concentration of molecules will be higher. So later in the process of the converting fuel from the algae which is used will contain lesser amount of the water. So the conversion process will be easy and cost efficient. So let us see how photobioreactor works. As we can see from the feeding vessel, the flow progresses to the diaphragm pump which moderates the flow of the algae into actual tube. Built into the pump is the carbon dioxide inlet valve. The photobioreactor itself is used to promote biological growth by controlling environmental parameters, including light. The tubes are made of acrylic and are designed to have light and dark intervals to enhance the growth rate. The photobioreactor has a built-in cleaning system that internally cleans the tube without stopping the production. After the algae have completed the flow through the photobioreactor, it passes back to the feeding vessel. As it progresses through the hoses, the oxygen sensor determines how much oxygen has built up in the plant, and this oxygen is released in the feeding vessel itself. It is also at this stage that the optical cell density sensor determines the harvesting rate. When the algae are ready for harvesting, they pass through the connected filtering system. This filter collects the algae that are ready for processing, while the remaining algae passes back to the feeding vessel and the flow continues. Now, now I will explain the second method of producing algae. So open pond is method or procedure to produce algae. Open pond basically consists of natural water in natural ponds or container. Most widely used approach involves tanks or raceway like ponds, etc. Open pond system use typically 1 foot deep ponds from about 1 acre to several acre in size, in which the algae are exposed to natural solar radiation, which they convert into biomass. Typically, the ponds are called raceway ponds because their shape resemble a racetrack. They often use paddle wheels or other water moving devices to keep the algae circulating. This kind of open pond system are very easy to use as compared to closed system, but major limitation of such schemes is evaporation. Since these systems are open in environment, so evaporation of water is easily done. So because of loss of the water, usage of water increased and wastage of water increased too. And another disadvantage is it will contain higher amount of the carbon dioxide, so that the concentration of molecules will be less, so that it will contain higher level of water. There are a lot of ways available using which fuel from the algae can be derived. One of the most common method used by most the people is described below. Similar process like this is also used into oil press. In this process, oil from the algae is extracted and more than 75% oil can be recovered from the algae. Another method is called hexane solvent. This is one of the efficient and effective method for extracting oil from algae. The efficiency of this method is more than 95% of oil which is available. Now we will discuss about challenges for synthetic biology. One of the major challenges this procedure is to find the fastest and efficient technique for the production of algae. Using synthetic biology, we can try to improve the technique. Another challenge is to find the efficient technique which can be used to convert algae to fuel, which can convert most of the lipid from the algae to fuel. Here, synthetic biology can also be used for the conversion.